My name is Vibeke. I'm working as a senior scientist at GlycoSpot, and today I would like to introduce the mini lab suitcase by GlycoSpot used for lipase activity measurements. I will be telling you what's inside the suitcase and the mini lab and using the corresponding test kits with it. I would like to start introducing the different elements of the mini lab by GlycoSpot suitcase, where we go in, and at the top we have a felt pocket which contains a certificate for the series which is in the suitcase, a cloth to wipe the cuvettes, which we use, as well as a protocol for the assay. At the top we have the mini lab by GlycoSpot lab bench, which contains different slots for the different parts you will need for your experiment. It contains a timer, and it contains some rows for cuvettes and other reagents, and where you can position your pipette. The positions for the different reagents are labeled A, B, C and 1 to 8, so you can follow your samples. The next element is the series, the spectrophotometer, which we use to measure our sample. It goes in the lower slot of the mini lab bench by GlycoSpot. The next element are the pipette tips, which go in this upper position. The pipette tips are used together with the fixed volume pipette, which has a holding position down at the lower corner here. The next element you will need is the rotator, which is used to incubate your samples. It also has the wings to hold your samples, which need to be attached on each of the 4 slots. And has a power supply here. The next thing I want to introduce is the test kits, which is labeled with the Lipase assay kit, as well as a QR code which you need for the assay. It contains the substrate tubes, as well as some empty cuvettes and a reagent buffer. Now we are ready to continue with our experiment. We will take out one substrate tube and place in a slot over here in row C, and we also will take 2 cuvettes, which we need, one for a sample and one for the reference sample. I will place the one for the reference sample in A8, so it's the first away from our sample. And then we will take out the reagent buffer which we need and place it in the slot down here designated for it. The mini lab by GlycoSpot also contains a slot for a waste container, which we can position here in this upper corner. Now we are ready to start our assay. We start by adding the reagent buffer to our substrate tube. And slowly taking up the buffer and adding to the substrate tube. And now we are ready to add our sample. We take a fresh pipette tip I've prepared a sample here, and I will add 4 times the fixed volume to the substrate tube. I will close the lid and gently shake our samples 4 to 5 times and position it in the rotator and start our timer. While our sample is incubating, we need to ready our instrument, our series. We start, it will have a green light for that it's on and has power, and then it will have a flashing Bluetooth so it can connect to your mobile device. Now we are ready to scan our QR code, which is positioned on the side of the box. So we push scan QR code and scan. Now it tells me to place the reference cuvette into the series. I've prepared a reference cuvette here, which I position with an arrow pointing towards this arrow in the series. Push it down onto the stop position and now say measure reference. And now it's ready for our sample. If you want to put in a sample ID for your sample which is incubating, you will tap sample ID and then write what you would like to call your sample and save it. And now your app is ready to measure as soon as your incubation is done. Our incubation is almost done. That's why I will ready a pipette tip again on our pipette. When the timer is has counted down, I will take out the sample, immediately transfer it to the cuvette and measure it, as the reaction will go on in the cuvette. Now I'm ready to measure and push it quite down and push measure. It says high lipase activity detected in this case, which gives you a red smiley.